This topic discusses the chemical properties of acids and bases. So this is quite a long topic compared to some of the others that you have seen. However, that just underlies the importance of understanding acids and bases and their chemical principles, because it's very important for the biochemistry that occurs in the interactions between biomolecules. So the learning outcomes are as indicated. So first of all, let's look at the definitions of what an acid is and what a base is. There's several different definitions in the chemistry literature, but we're going to look at it in terms of as an acid as a proton donor and base as a proton acceptor. So we talked about the ionization of water and how it can generate protons or hydronium ion and hydroxide ion. So in this case, water could be considered an acid because it can give up a proton, and the hydroxide ion could accept a proton in the form of a water molecule, so it can be considered a base. So this is the Lowry-Bronsted or Bronsted-Lowry definition, which is the most useful in thinking about the structure and interactions of biological molecules. So that's the definition I want you to know. Also, there's descriptive properties of acids and bases. What makes a strong acid and what makes a weak acid? A strong acid basically will dissociate completely, and the concentration of the acid is equal to the concentration of the protons that will be present in solution, because the dissociation of the acid is complete. A weak acid, if you were to add to a solution, will dissociate only partially, and so we considered anything that's not a strong acid to be a weak acid. If it doesn't dissociate 100%, theoretically, we will call it a weak acid. There's another term that we're going to introduce, the acid dissociation constant. It is an equilibrium constant, but we give it a special designation as Ka. So it's a dissociation constant for an acid, and that's why the subscript is A. It's sometimes in chemistry you think of Ka as an association constant, but in terms of biochemistry, the Ka is reserved for acid dissociation constant. So keep that in mind, that the Ka is acid dissociation constant, an equilibrium constant. Since acids and bases are related based on whether the proton is being donated or accepted, we can come up with corresponding terms for bases as well, whether it's a strong base or a weak base, or the association of a base with a proton, but we're not going to do that here. What we're going to do is think of everything in terms of an acid, how it can dissociate to generate a proton in aqueous solution. So we're going to focus on the acid component here in this course. So when we look at an acid dissociating, we'll often abbreviate it as HA, and the proton of the acid will dissociate and interact with a water molecule to form a hydronium ion, and the negatively charged species after proton dissociation from the acid, and we define these terms. And so the HA is the acid, it donates the proton. The base accepts the proton. In this case, water will be accepting the proton to form the hydronium ion. The hydronium ion can lose a proton if it goes back in the other direction to form water, and so we call this the conjugate acid. And then the acid that dissociated to form the proton to interact with water to form the hydronium ion forms a negatively charged anion called the conjugate base. So the acid, when it dissociates, will form a proton in the conjugate base. The proton is picked up by a water molecule to form the hydronium ion. So this is how acids donate protons in aqueous solutions. Since the water molecule is here and we know its concentration doesn't change, it's 55.5 molar, it's often removed for simplicity, and we'll just look at the acid as HA and looking at its donation as a proton and the formation of its conjugate base. But remember, there's a water molecule here that's accepting that proton to form the hydronium ion. We can look at a base in terms of a base accepting a proton. So a base will be present in solution, get a proton from water to form the conjugate acid, and the hydroxide ion will be formed in the process of that proton dissociation of water. And we can abbreviate that shown here. But we will not be using this convention. We're going to focus on this one and think of everything in terms of an acid in dissociating a proton into the aqueous solution to form the conjugate base. So I just wanted to show this for completeness, but we're going to focus on all biological molecules as acids. It just keeps things simple to focus it on that way, and you can understand the concepts better. So we mentioned strong and weak acids before. So a strong acid dissociates almost completely. Nothing's 100% complete in any biological system or any type of chemical equilibrium environment. So the strong acid, like HCl, where you take chloride gas, bubble it through water, some of the chloride gas will form chloride ion, and you'll end up getting a very strong acid, hydrogen chloride, or basically hydrochloric acid, and that dissociates into protons and chloride ion, and so it's a very strong acid. This arrow here is pretty much very big, and the reverse arrow is very small. It's pretty much 100% dissociate. So if your hydrochloric acid concentration is 1 molar, you have 1 molar of protons in that solution. The Ka is the acid dissociation constant, so we can write this expression out in terms of products over reactants. So that would be shown here for the strong acid, would be the concentration of the protons times the cross-related chloride ion over the concentration of the hydrochloric acid itself. And if we look this value up for HCl, the Ka is 1 times 10 to the minus 6 molar. And so you recall that when we have brackets, they're all molar concentrations. So like I said, just think of everything in molar. It'll just make your life easier for these calculations. And so this is saying that there's very little HCl present, because this is over 1. So it's 1 times 10 to the minus 6 over 1. So that's 1,000,000 to 1 difference in terms of for every 1,000,000 protons, we have 1 HCl molecule. It's not 0, otherwise this would be, Ka would be infinity. But there is a little bit around, but there's not much. Since this Ka is so large, we basically will say that for our purposes of our calculations, that the concentration of the hydrochloric acid is equal to the proton concentration in solution. This is for only strong acids like HCl, sulfuric acid, nitric acid. Everything else that we'll be looking at will be pretty much weak acids. So weak acids do not dissociate very much in water. So let's look at an example of acetic acid. So acetic acid is a weak acid, and it dissociates to form the hydronium ion, or the proton in this case, and the conjugate base, the acetate ion. So when we look at the value for the Ka, the acid dissociation constant, concentration of products over reactants, and look that up in the table, we find that it's 1.74 times 10 to the minus 5 molar. So that means this is over 1. So there's actually a lot more of the acetic acid present in solution than there is of the conjugate base or the proton. So most of the acid exists in the acidic form. This is much different than it was for hydrochloric acid. So this Ka value tells you something about the strength of an acid. If the Ka is greater than 1, we can consider it a strong acid. If the Ka is less than 1, we can consider it a weak acid. And some acids are stronger than others, and some acids are weaker than others. The Ka will tell us that. But this is a general rule if you want to think of, is it a strong acid or weak acid? Greater than 1, strong, less than 1, weak. And then the values there will be either stronger, strong acids or weaker, weak acids. So why is it important to understand weak acids and weak bases? Because many biological molecules, compounds involved in biochemistry, are pretty much weak acids. And examples would be amino acids, DNA, RNA. So the A and DNA and the A and RNA refer to acids. So here's a amino acid, and we'll be talking about amino acids in the context of our first macro molecule proteins. And the amino acids are basically have this type of structure where you have a carboxylate group that's similar to that acetate conjugate base that was formed from acetic acid dissociation of a proton. And that's a weak acid, just like acetic acid's a weak acid. And the amino group is, in terms of its ability to, in this case, accept protons, is a weak base. So biological compounds typically have weak acidic or weak basic groups on them. There are some exceptions, like those that have sulfur as a part of a hydrophilic group, as we saw with the lipids in the topic where we looked at the hydrophobic interaction. That basically is like sulfuric acid in terms of a strong acid group, so we don't think of it being protonated. But most of the time, we're going to have weak acid or weak basic groups. So it's important to understand how we can measure these species under different conditions in the cell or other environmental conditions in which the biochemistry is occurring. So we need to look at a way to measure the amount of these acidic and basic groups, whether it's protonated or whether it's deprotonated, and that's going to depend upon the pH. The pH of the solution is going to dictate whether these groups are protonated or deprotonated. And these charges make a big difference. For example, we've shown that how water molecules interact with a negative charge versus a positive charge. If you were to eliminate the charge on this, water molecules are going to interact differently. If you were to eliminate the charge on this group, say it were interacting with a positively charged group of another molecule through a non-covalent ionic association, protonation of this group would make it neutral, and you break up that ionic association. So there are huge ramifications if the pH changes too much with these biomolecules, because you start altering their structure, and in macromolecules you start altering their function. And I'll show some examples of that later in this topic. So we need to have the pH in cells and in biological environments stay constant in order for the enzymes and the biological molecules to do their work. So we have what are called buffers, which are weak acids or bases that are present that maintain the pH of a solution. So there's another topic on buffers, and so you can listen to that topic. But right now we're just going to focus on trying to characterize these acidic and basic properties of these functional groups on biomolecules. So the best way to do that is to consider an example. So we'll look at the weak acid acetic acid, and recall that basically I'm drawing the structure out now, showing you the dissociation of the proton. So this is to emphasize the charges. This is going to form a covalent bond here, and so you're not going to have charge, but if you think of them as ions that are interacting, it's just dissociating into the proton plus its conjugate base. And so we can write this as a equilibrium expression of the acid dissociation constant Ka, or the concentration of the proton over the conjugate base, over the concentration of the acid that's present. And we're going to rearrange this equilibrium expression, and we're going to solve for the hydronium ion concentration of the proton concentration. And we end up with this expression. Then we're going to take the log of both sides. So the log of the hydronium ion concentration equals the log of the Ka. And remember logarithm properties, we can rewrite this as plus the log of the acetic acid, the acid species over the conjugate base. Okay, we're going to further change this equation, and we're going to take the minus 1, multiply both sides of the equation by minus 1, so we get minus the log of the proton concentration equals the minus the log of the Ka, and minus the log of the concentration of the acid over the concentration of the conjugate base. And we're going to rename these because this little p here as part of pH is the minus the log of the base 10. We don't have the 10 here, but that's what log is, the base 10. So that becomes pH. This becomes pKa, and we could just leave this as negative, but negative signs have a tendency to float away. So if we just change the numerator and denominator here, that changes the sign of this log from negative to positive. Again, that's logarithm properties. And so then we have the log of the conjugate base over the acid, and we end up with this new expression here, which is called the Henderson-Hasselbalch equation, where the pH equals the pKa plus the log of the conjugate concentration of the conjugate base over the acid. Again, this is at equilibrium. So this is a very useful equation in which that we can take a value of a buffer, and knowing its pKa and what pH of the solution that we have, we know the ratio of the conjugate base over the acid, and we can calculate those species as well. So it's important to understand biological molecules as it relates to this interaction between functional group pKa's and the pH of the solution. So the really important take-home point here is the pKa of the acid is going to let you know at how good is that acid at deprotonating, and at a given pH, is that acid going to be present as the acid form or the conjugate base form, and whether it's going to be charged or not. So different parts of an organism have different pH values. Cells are in different pH environments. And so it's important to understand how these functional groups behave under different pH conditions. So if we look at this ratio here, it's informative in terms of letting us know about the amount of conjugate base and acid forms. If we have equal amounts of conjugate acid or base, then this becomes the log of 1, which is 0, then the pH equals to pKa. So for me, if I know that the pH equals to pKa, then the concentration of the acid and the conjugate base are equal. If the pH is below the pKa, you're going to favor the protonated form, the acid form. If the pH is above the pKa, you're going to favor the deprotonated or the conjugate base form. So you'll see and understand this better as we go through and practice problems. So here's a table showing some examples of weak acids, and you'll see the empirical formula, the name, and the conjugate base form, the pKa value and the Ka value. So you can see the Ka values usually have negative exponents here, and then the coefficient is shown here in terms of the precision based on the measurement, and then the pKa. So that's why we keep it as pKa. We don't have to worry about 1 times 10 to the minus 4 or 5 or 13. It's just easier to keep track of the pKa value. So the stronger the acid, the lower the pKa. The weaker the acid, the higher the pKa. And you can see that in the dissociation constant, acid dissociation constant. Ka is smaller, therefore the pKa is larger because of the minus log aspect of determining the pKa. So the key thing to remember here, you can rank these based on the strongest acids of the weak acids is the one with the lowest pKa, and the weakest is the one with the highest pKa. You don't have to memorize these numbers, just know how to use them. So how are these pK or Ka values experimentally determined? Well, you do titrations of molecules to figure out these pK values. And so here's our acetic acid situation, once again, looking at the deprotonation of that. And what we're going to do experimentally is we're going to weigh a certain amount of acetic acid in a certain volume of water, and we'll put it in there, and that's going to be our starting point. And we're going to stick a pH electrode in there, and it's going to be starting here. We haven't added any base. And now we're going to titrate that acid by adding hydroxide ion, equivalents of hydroxide ion. And so this would be in terms of moles, mole equivalents. And then we'll have a pH value here with a pH meter. And so all of you have done this at some point in a chemistry course where you've added base to an acidic solution. You may have not had a pH electrode in there to measure the pH at every step, but you had some kind of indicator, like phenolphthalein, that would turn pink once you reach the equivalence point. And basically, phenolphthalein is a weak acid, like acetic acid, that once you have completely titrated the strong acid that was present, usually you're doing HCl or something like that, and you're going to use all those protons up first, and then you'll start to pull the protons, have the protons dissociate from the weak acid and shift this equilibrium, and eventually this will be the colored species. So if you get close to that equivalence point, you'll see a pink color come, then it'll go back. So you know you're close, and then usually one more drop, boom, you're done. And you've now completely converted the acidic form of your indicator to the conjugate base form, which is recognized color. And so it's a chromophoric species when it's deprotonated. And so that's how you found your endpoint. But when we have something like acetic acid, it's a weak acid. So an indicator wouldn't be useful here. So we have to use a pH meter to monitor the pH changes. And so when you do that, you usually see a curve that looks like this. It'll start to go up as we add equivalents of hydroxide ion. And then at the half equivalence point, where we basically have titrated half the acid, that's where the concentration of the conjugate base equals the concentration of the acid. So the pH will, at this point, is at the half equivalence point, that's going to be the pKa. That's when you have equal amounts of conjugate acid and the conjugate base and the acid form. That's the 50-50 mark, half equivalence, titrated. So you extrapolate your line over here to the y-axis, and that will be your pKa of your acid, in this case the pKa of acetic acid. You can continue to titrate. pH doesn't change very much in these regions until you start to get towards the end point, and then the pH will rise rapidly as you add the final equivalence of base to completely titrate the acid. So this titration curve of a weak acid will look something like this for one titratable group, where it'll go up, level off, and go up again. The leveling off inflection point occurs at the half equivalence point. Notice the pH does not change very much in this region here, and so if we were to extend it, these regions, in terms of a buffer, if you will, the buffer keeps the pH from changing, and so that's usually plus or minus 1 pH unit above or below the pKa value. So if this is a pKa of 4.8, you go down to 3.8, yeah, the pH changed a little bit there, but over here it's more stable. And so I usually like using the value of plus or minus a half pH unit, not plus or minus 1 unit. 1 unit's a little too far down. The pH is kind of distance, but 4.3 point, that's a little too far away. I like to keep it plus or minus a half. It's more buffering ability there, because you can see you add a little bit of acid, a little bit of base, you can move above and below this equivalence point, and the pH is not changing. I mean, you can see that. You can add acid to move down. It takes a while to really see the pH meter start to make readings below the pKa. Same thing with the pH above the pKa value. You add more base, you see it's not changing very much until you hit a certain equivalence point, and then it'll go up. So if you want this pH to be stable, put a weak acid in there, and if you can find a weak acid that has a pKa equal to the pH of your solution, you'll be right here, and you'll be able to accommodate any acids or bases that may form because of a biochemical reaction catalyzed by an enzyme, so you won't have pH changes. And so that's what a buffer does. It prevents the pH from changing a lot. And the ideal buffer will be one in which the pKa of that buffer equals the pH of the solution that you're interested in. We'll be talking more about buffers in a different topic, but wanted to kind of show you the properties of what makes a buffer. It keeps the pH from changing. It's a weak acid or it's a weak base. Let's look at a situation in which we have an acid with multiple titratable hydrogens. So phosphoric acid has 3 ionizable protons. So if you were to do a titration curve determination of phosphoric acid, you would have this type of curve, 3 plateaus, 3 steps. Each midpoint of that step would be a pKa value. So here's shown phosphate completely protonated as phosphoric acid. The first titratable group has a pKa of 2.2, the second one 7.2, the third one 12.7. So each has its own buffering capacity, and once you take the first one off, which is can dissociate quite readily, the second one's a little more difficult to completely remove. The third one's a lot more difficult, and that's because as you start titrating protons from phosphoric acid, you're putting negative charge on the resulting species of the conjugate base. And so that first titratable proton comes off rather readily to form the -1 conjugate base form. Taking the second one off is a little more difficult, but once you do that, now you have 2 negative charges on the conjugate base. Now you're trying to introduce another negative charge on the molecule, and they're all close in proximity, and so as you try to put more negative charge on that, the electrons are starting to get too close into space because you have these extra electrons. So it really doesn't want to have 3 negative charges in close space. And so that's why that last one's a lot more difficult. When you start off, there's no negative charge. That first one comes off quite readily. The second one, not too bad, but that third one's tough to get off because now you have all this negative charge. You're trying to induce more negative charge on the molecule, and the orbitals of the sigma bonds and their spatial arrangements are such that it's harder to accommodate this negative charge. It's less thermodynamically favorable, and therefore the pK of this last titratable hydrogen is much larger than the other two. We can also map the species as we go through the titration. So this is a way to look at the species during the titration course of going through the pH changes here. And so you can see we start off at low pH, everything's protonated. We start losing this first species here, and there's less and less of it. When we get to the equivalence point at 50%, we have 50% of the protonated form, 50% of the deprotonated form, or the conjugate base form. And that's where it'll intersect right here. This is your pKa. As you continue titrating, this species will go to 0 basically, and now it'll be all in the deprotonated form of the conjugate base. And then we start working, that's the equivalent point of 1. Then we start to work down again to the next equivalence point, and now we start to form the doubly charged negative ionic species of the conjugate base until we hit the next equivalence point where it's 50% of the -1 and 50% of the -2. Then we continue to titrate, to completely titrate that second proton off. And then we begin the third proton titration to form the triply negatively charged species. And at the 50%, we are at the third equivalence point and get our pKa value for that third titratable group. And then we can continue on to completely titrate that for the rest of the titration curve. So keep in mind the processes are involved of the titration curve, what's happening. You'll need to understand the titration curve. You'll need to understand how to generate a titration curve. Again, at the equivalence points, the halfway point between the equal concentrations of the acid and the conjugate base. Those will be your pKa values where you have these plateaus, and the pH equals to pKa. When you're at the equivalence point, you completely titrate that first group, so you shoot up and start titrating the second group. Second equivalence point, shoot up, third group. So these halfway points, 50-50, 50-50, 50-50, for each titratable group is always going to be at the half equivalence point, and the whole values will be complete equivalent. So you do the first titration, the second titration, the third titratable group. So there's problem sets to practice this, but this is what you should be able to recognize and understand or how do you get pKa values from these plots? How do you sketch a plot knowing the pKa values of a particular biomolecule, and what points here that you have on the x-axis in terms of equivalence of base, in terms of being the 50-50 positioning of the conjugate acid and base forms, the acid and conjugate base forms versus the completely titrated acid into the conjugate base. So far, I've shown you some examples of a molecule with one titratable group. That was an acid, and an acid that had three titratable groups. But those are both acidic functional groups for those molecules. But a lot of biological molecules have both acidic and basic groups attached to the same molecule. And so we call these molecules ampholytes. They have both acidic and basic groups, basic pKa's. One of the large classes of biomolecules that have these features are amino acids, and that's shown here. Showed that a little bit earlier in this topic. So we call these amino acids because they have an acidic group or carboxylate, similar to our acetate, acetic acid, and we have a basic group, an amine. And so we have acidic group and basic group. So this is an ampholyte. And so we can titrate these molecules to figure out the pKa's of these acidic groups. Considering an amino group is basic, but we can still consider it an acid as if it dissociates. So we can get a pKa for that as well. But it's basic in that it wants to keep the proton as opposed to wanting to give it up, compared to what we would consider for the carboxylate here, for the carboxylic acid group. So we consider it as basic, but it can also be shown and treated as an acid and get a pKa value for it. So let's look at the titration of this particular molecule here. This is actually glycine, the amino acid glycine. The R group here varies, and so there's 20 different amino acids that you'll have to know and recognize and be able to draw. And the R group's different, everything else is the same. And so glycine here just has a hydrogen on this alpha carbon. And so that's what we're looking at here. So if we completely protonate this molecule, you see that the amino group is positively charged, the carboxylate is now neutral, and so the overall charge of this molecule at low pH is 1. The pK of this carboxylate is 2.3, so if the pH were 2.3, we would have 50% of this being protonated like this, 50% would be deprotonated like this. If the pH were below the pKa, we'd have more of this protonated form versus the deprotonated form. If the pH were above the pKa, we would have more of the dissociated negative ion conjugate base form in the acidic form. The charge on this molecule, if the pH is below the pKa, will be +1 overall. There'll be a combination of some of this dissociating, but the predominant population will be +1. As we increase the pH, we start to add base, do a titration basically, and this proton is going to be more freely dissociated, and eventually it'll be dissociated completely under one equivalence point of base, and then we will have a species in which there's a positive charge on the amino group and a negative charge on the carboxylate group, and so that molecule is electrically neutral. So it's overall electrically neutral, but part of it is positively charged and part of it's negatively charged. And so we call this a zwitterion. It contains both positive charges and negative charges, but when you look at the overall charge on the molecule, it's electrically neutral. Low pH, it's cationic. At the right pH, when you have this equal balance between the dissociation of the carboxylate but not losing any protons on the amino group, you have a zwitterionic form with a net charge of 0. If we keep adding base and keep increasing the pH, we'll start to titrate this other proton on the amino group, and that becomes now a lone pair of electrons on there because it lost its proton, and now it becomes the anionic form at high pH and becomes -1. The pK of the amine is 9.6. So if the pH were 9.6, we would have equal concentrations of the protonated and deprotonated form. So like I said, even though this is a basic group, we can still think of it as an acid, and we will think about it in terms of a pKa value of how it gives it up. So that's why I mentioned earlier, think about things in terms of acids. But if the pH equals the pKa, we know it's 50-50, and so half of it would be deprotonated, half of it would be protonated. Keep going higher with the base, keep going higher in the pH, and you'll completely deprotonate it. You'll be left with the fully anionic species. So the zwitterion is important to understand where that occurs because it's electrically neutral. And the pH at which this average charge sums to 0 is going to be where the zwitterion exists. So the pH in which the molecule is a zwitterion is called the isoelectric point, or the PI. It's very important to know that because it has consequences in how macromolecules function and how soluble they are. And we'll show some examples of that in this topic. So know what an amphiolite is, know what a zwitterion is, and what the isoelectric point is. So let's look at the titration curve for the amphiolite glycine. So recall the completely protonated form of the molecule at low pH will be a +1 charge. Then we titrate the carboxylate hydrogen, the carboxylic acid, a proton, and then we form the carboxylate. This is the zwitterion at 0, and then we continue with the titration of the proton off of the amino group to get the neutral version of that functional group, making the overall charge on the molecule -1. So let's take a look at how this is occurring. So if you look at the titration curve shown here, we'll have 2 pKa, so we have 2 levels. So again, shown here, the levels aren't as flat as my hand-drawn ones, but the idea is the same. There's an inflection point where the half equivalence point is for the first pKa, and then the next half equivalence point for the titration of the second group is at 1.5, so there's your pKa2. And then if we look at the molecule as the +1 charge, that's shown here. So that's at low pH, basically pH 0. And then we start titrating that carboxylate group at the half equivalence point, and then we completely titrate it away. And so we have the zwitterionic species at this point, and that occurs at the completely titrated carboxylic acid group at this midway point before we start to titrate the amino group. And so at this equivalence, full equivalence point of this first group that was titrated, we extrapolate back over to the y-axis at the pH, and that's going to be the PI. And we start titrating the second group. We get to its half equivalence point, 50% of the conjugate base, 50% of the acid form. That'll be our second pKa, and then we continue up to the full second equivalence point, and that's when we have the completely negatively charged species at -1. So the pi, the zwitterionic form, is shown here. So if you look at the pi value and you see where it's located, it's basically the average between the two pKa values. That's how the pi value can be attained from the pKa values. But you have to know where the zwitterionic species is happening in order to calculate the pi. So in this case, we have two titratable groups, two groups that allow the formation of a zwitterionic species, and you have a pi. Compare this titration curve to the one we had for phosphoric acid previously in this topic. In that case, we had 3 pKa values, but there is no zwitterionic form, an equal number of positive and negative charges on that molecule. So there is no pi existing for that compound. So the pi is something that has to be generated by the molecule that has functional groups that allows it to be electrically neutral but have an equal number of positive and negative charges. That's the key thing. It has to have an equal number of positive and negative charges. So this is the titration curve for the ampholyte glycine. If we look at the different species that arise during the pH titration curve changes, at low pH, it's going to be pretty much the protonated form, half equivalence point, we're at 50% protonated, 50% deprotonated for the carboxylic acid group. Then we finish the titration to the complete equivalence point, where we're all in the zwitterionic form, and then we begin to titrate the amino group, and when you get to 50% of it titrated as the protonated and deprotonated forms, you reach your second pKa, and then you continue to complete the titration of that functional group to the final second equivalence point. So this is very similar to what you've seen shown for phosphoric acid. So the pi is at the pH in which the zwitterionic form is available, and if you look at the any situation in which you know the pi of the zwitterionic form, if the pH is above the pi, you're going to have more of the negative species present. If the pH is below the pi, you're going to have more of the positive species that's present. So this is very useful to know what the pi of a molecule is, because if the pi is equal to the pH, the molecule is electrically neutral. This is zwitterionic form. If the pH is above the pi, the molecule is going to have an overall negative charge. If the pH is below the pi, the molecule is going to have an overall average of a positive charge. So it's important to know if a molecule has a pi or not. Let's look at a situation in which we have more than 2 pKa's, because in terms of glycine, the pi was the average of the 2 pKa values. So we can mathematically calculate that one. Don't necessarily need to generate it off the titration curve. But how do we look at pi if we have more than 2 ionizable groups? So let's look at another amino acid called aspartic acid. So aspartic acid is has similar features to glycine. You have a carboxylic acid group off the alpha hydrogen. You have an amino group off the alpha hydrogen. But all amino acids have these two functional groups, but the side chain is different. So in the side chain of glycine, this was just a hydrogen on this side chain. Here, in this case, it's a CH2COOH, so it has another carboxylic acid group. So you have 3 titratable protons. So now it's 3 like phosphoric acid. So you expect to have 3 pKa values. If you titrate these, that's indeed what you have. Even though these are 2 carboxylate groups, they have different pKa values. The first pKa value is based on titrating the alpha carboxylic acid group. The second pKa is based on the titration of the side chain carboxylic acid group. So how do we know this? Well, based on the formation of the negative charge on the carboxylate due to the titration, it's more stabilized than the side chain because it's closer to this amino group in terms of bond length. So for example, this carbon is 1 bond, this is 2 bonds away from this protonated nitrogen, whereas this carboxylate is 1, 2, 3 bonds. It's further away. So the positive charge that helps stabilize these electrons on the carboxylate is further away for the side chain compared to the alpha amino group. Therefore, this first carboxylate is more acidic. Its conjugated base form is more stabilized, and hence its pKa is lower. The third pKa would be the amino group, and that has a pKa of 9.8. Shown here is the lone pair of electrons. Sometimes they're drawn in, sometimes they're not. You have to be aware that they're there, and that's where the proton can be attached. So if we look at the charges at the equivalence points starting at low pH, and after you do the complete titration of each one of these acidic groups, the net charge in this case is a +1. Then we have a zwitterionic species here. Then we have a -1 after we completely titrate the second group. And then we titrate the amino group and we have -2. So these are the charged species at the end of the equivalence points. So let's look at the titration curve for this species. Oh, just before we go though, where it's electrically neutral is right here at this pH after the first equivalence point, because you have equal number of positive and negative charges. So there's always going to be an isoelectric point at a species, isoelectric species at some point in the titration of amino acids. Key thing is to recognize what the molecule looks like when it's protonated or not, and whether there's a corresponding charge. Some groups, like the carboxylates, will become negatively charged when deprotonated. They're originally neutral. Groups like the amino groups are positively charged and become neutral once they're titrated away. So you have to kind of know the structures and understand their acidic and basic properties in order to determine if there's a positive or negative charge on a group at a given pH, or neutral. So let's take a look at the titration curve of the amino acid aspartic acid. So shown above are the different chemical structures of aspartic acid as it goes through a series of deprotonation steps. The arrows point to the location in the titration curve in which the majority of the species exists. The moles of the equivalent hydroxide ion added per mole of aspartic acid is on the x-axis, the pH is on y-axis, and we can look at a titration curve and determine the pKa value. So at half equivalence points is here. The plot is a little off in terms of how it was rendered, but the pKa of the first titratable group is 2.1. 1.5 equivalence point, pKa is 3.9, and 2.5 equivalence point, the third pKa is 9.8. And looking at the species after a complete titration of the first acidic group is where the zwitterionic species exists, and so the pH at that point is the pI of this molecule. So that's how you can determine the pI from a titration curve. It's no different than the example I showed before. However, there's 3 pKa's involved. Mathematically, we can determine the pKa from, the pI from 2 pKa values that flank the pI value. So it would be the average of this pKa and this pKa, which would be 4 plus 2 is 6, divided by 2 is 3. So the pI is 3 in this case, an average of the 2 pKa's that flank the zwitterolinic species. You don't average all 3. If you're just guessing, you'd have to guess 2 sequential ones. So in this case, you would have either the first 2 or the last 2. But I don't want you to guess, I want you to figure it out. So as long as you know where the zwitterolinic species is, whether it's at the first equivalence point or the second equivalence point or the third, depending on how many titratable groups you have on the molecule, the pI is going to be the average of the 2 flanking pKa's. Now, I'll show more examples of this when we talk about more complex molecules that have more than 3 titratable groups, like peptides have 4 or 5 or 6, or proteins that can have 20 or 30 different groups. I'll show some examples in this topic of the ramifications of pH and how it affects protein and macromolecular interactions. But for you, practice the problem sets and the acid-base posted problem sets, as well as some of the other ones that we'll see in other topics, because we'll be doing more titrations. Understand the key features of a titration curve, defining where pKa's are and pI's are. So let's look at an example of a molecule that has a lot of titratable groups. Proteins are polymers of 20 different amino acids. And so proteins can be hundreds, 500 amino acids or more. And so there's many titratable groups that are present in these side chains, and hence you have a lot of acidic and basic groups that can give rise to different electrostatic features on the surface of a protein. So let's look at the example of ubiquitin at 8 different pH values. And shown here is the surface potential in terms of color. Blue color indicates amines, things that are protonated and positively charged. The red would indicate like carboxylic acid groups that would be on, for example, aspartic acid side chain. That would be deprotonated and negatively charged, which would be red. Then you have some side chains of amino acids that have no titratable groups. They're aliphatic in nature, they're hydrophobic. So it doesn't matter what the pH is, they don't pick up a charge or lose a charge or become neutral. They stay neutral all the time, and they're shown in white. So you can see if we have a low pH of 4, many things are protonated. Some things are still deprotonated because those groups have pKa's that are below 4, and so they've been titrated away and are negatively charged. As we increase the pH, think about adding hydroxide ion equivalents and start to titrate some of the groups that were charged or electrically neutral, you start to see changes in the surface potential as you go to higher pH. So compare pH 4 to pH 11. Pretty much mostly blue with a little red. And at pH 11, it's all red. So you can see the effect of changing the pH of a solution on the protein's overall electrostatic features. So obviously this is a different molecule than this one because this is more positively charged, this is more negatively charged. So we can look at transitions at different pHs. And so shown here is if we go from 4 to 5, the charge surface went from +6 to 1. At 6 to 7, we're transitioning to pH 8, the zwitterionic species, and then we start to get more anionic as the pH goes up. So with all these ionizable groups, it's difficult to calculate the pI, but it can be done. But the best way to do it would be just to do it experimentally, because some groups that may be acidic or basic are buried on the inside of the protein, so they won't be able to be titrated away. And so you have to kind of know what groups are on the surface in order to be affected by the pH, in order to figure out what the pI is. And so it's hard to calculate it from purely knowing the amino acid residues that are in the protein sequence. So what happens is, experimentally, the protein is allowed to migrate in an electric field from a positive pole to a negative pole, and when it starts to change its charge state and lose positive charge, its migration to the negative pole slows down to a point where when it becomes electrically neutral, it stops. And so at that position, we know that the pH at that position is the pI of the protein. So in this case of ubiquitin, the isoelectric point is 6.8. So we know at that point it's electrically neutral. If the pH is below the pI, the molecules will be more positive. And the further away from the pI, the more positive it will be. And if it's above the pI, it's going to be negative. And the further away is from the pI, the more negative it will be. So keep that in mind. The pI is where it's electrically neutral, below more positive, above more negative in terms of the pH of the solution. So this surface charge is important because we need charges to help these molecules to be soluble. So DNA is an example of a macromolecular molecule that is negatively charged. It's got a lot of phosphates connecting the nucleotides bases, and so it's very acidic. That's hence in the name, deoxyribonucleic acid. And so it's very negatively charged. Because they're so negatively charged, these DNA molecules will not interact with one another because they're all negative. So there's some repulsion there, so they will not aggregate. We take advantage of these features in that a protein can be positively charged, and because it's positively charged, it can interact with the negative charges of the DNA to promote a protein-DNA interaction. And so proteins that are involved in gene regulation or turning on a gene that bind to specific promoters based on the base pairings are initially attracted and help to form the complex through the interactions of the positive charges on the protein with the negative phosphate backbone. And then specific amino acid residues on the protein themselves will intensify that interaction through base pair recognition. But the initial encounter is through this positive and negative charges that are present on the molecules. Protein solubility is also affected by pH. So here's a different protein called beta-lactamase, and its surface potential will be different at different pHs as well. So if we have the pH of the solution greater than the pI for beta-lactamase, the molecule is going to be more negatively charged, as shown here. And this is just different rotations of the molecule. And its overall negative charge will keep the molecule from interacting. Sure, there's some positive charges here, but there's only a couple of them, most of it's negative. So it's going to kind of keep the molecules separated, just like the DNA example. If the pH equals to pi, now each molecule has an equal number of positive and negative charges. The distribution on the surface is maybe a little polarized in that there's more negative on this side, maybe a little more positive on that side. So these things will now start to clump together, and the negative surfaces will interact with the positive surfaces on another molecule, and they'll tend to aggregate. So the protein is less soluble because of these charges being neutralized on individual molecules to be equal number of positive and negative and promoting their interaction. If we lower the pH further to put more positive charges on the molecule, it has the same effect as we make the molecule more negative. They'll repel each other and they won't aggregate. So if we were to graph the protein solubility as a function of pH, you can see that the proteins are very soluble when the pH is well below the pi value or well above the pi value. But as we creep towards the pi value with the pH, protein solubility is lessened to a point where it could aggregate and crash out of solution. So if you're doing protein purification, you do not want the pH of your solution to be at the pi of your protein. You want at least 1 unit above the pi. So if the pi here is 5.4, you want to be at 6.4 to be very soluble, or 4.4 to be very soluble. So that's a rule of thumb to keep the protein soluble so they don't aggregate when they become zwitterionic. Another way to help proteins remain soluble is to increase the ionic strength, to add some salts into the solution. So we already demonstrated that pH is important, so you're not close to that pi value, and so you want the pH to remain stable at least 1 unit above or below the pi value. Another important component will be the ionic strength. How much salt do you have in the solution? How much sodium chloride is in there, or other types of salts? So the example is shown here. Say we have one protein in our solution, because a lot of times we have multiple proteins present in biochemical reactions, and so we need to study some mechanisms and some pathways with multiple proteins. So you can imagine if you have a positively charged protein at a given pH and a more negatively charged protein at the same pH, because they have different compositions, they'll have a tendency to interact with one another, and they may crash together and come out of solution. So one way to help solubilize them is to add, say, sodium chloride, positive and negative ions, salts into the solution. And so what will happen is that your positively charged protein will be surrounded by the negatively charged chloride ions, say in sodium chloride, to form this type of counter ion sphere. The negatively charged protein would have the sodium counter ion sphere. And there's 1 or 2 little sodium or chloride ions that kind of infiltrate this counter ion sphere around the protein's environment, but it's overall going to help shield the charge of this protein that's positively charged with the negatively charged protein due to this counter ion atmosphere. So it makes this protein less positive. This counter ion sphere makes this protein less negative, so it'll tend to disrupt the interaction and make it weaker. Remember that each one of these ions, if it's a sodium ion or potassium ion, is also surrounded by water molecules as well. Proteins are also surrounded by water molecules, so there's water around this situation as well. So keep that in mind too. It's just not the ions themselves. This is not a vacuum, but this is an aqueous solution. So you do have water molecules still surrounding each one of these chloride or sodium ions, as well as water around the protein as well. So water is also possible interacting here as well. So one way to help solubilize different species present in a solution, in addition to pH, is ionic strength. So there's a limit here, because if you add too much salt in there, now you're starting to affect the amount of water that's available for certain things, and you have too much salt, so the water starts to run more of the salt, and you can have the opposite effect to disrupt and cause things to crash out of solution, because there's not enough water to go around to provide proper hydration. So there's this balance that you need to maintain in terms of ionic strength and pH in order for biomolecules to stay in solution and not be affected by all the charges and differences in electrostatic potential based on these acidic and basic groups. So the last few slides showed examples of the importance of pH and how this affects the acidic and basic functional groups on macromolecules, and the things we need to consider when we're looking at biomolecules in solution. So practice on the acid-bases problem set and work out those solutions, work on the mastering problems, and it'll become clearer as you start to think about these in problem solving.